Thank you very much. My name is Savanka Pal. I'm a clinical neurologist and a professor of neurodegenerative disorders and clinical trials at the University of Edinburgh in the United Kingdom. It's very good to be here today and give an update about the MND-SMART trial which we're running. So, a very brief background about motor neuron disease. We know that this is not such a rare condition. If you think about the lifetime risk, 1 in 300 people will be affected by this disease in their lifetime, and it affects people throughout the whole of their adult life. More commonly in people's 50s and 60s, but can occur in any age group. And it affects people by causing weakness of the arms and legs, speech and swallowing difficulties, and breathing problems. And up to half of people with the condition will have memory and behavior changes, and 15% of people will have dementia. It's unfortunately still a very difficult disease to diagnose. It takes 1 year from the first symptoms to when a definitive diagnosis is made, and it's a very rapidly progressive condition. After diagnosis, the average survival is only 2 to 3 years, and the reality is that 1 in 3 people will die within the first year. But against that background, there's only one licensed drug that has been approved globally, Riluzole, that was approved in almost 30 years ago now. So the challenge for us as a community is to think about how we improve outcomes for people with new treatments and identify these through clinical trials. Also, how we discover better ways of making an early and accurate diagnosis, either through blood-based markers or digital biomarkers. And all of this requires greater participation of people in research. So we set about doing this really by creating a team, an interdisciplinary team involving people living with motor neuron disease, ALS, neurodegenerative disorders scientists, people who have had expertise in clinical trials with medicines that have improved outcomes, for example in cancer, using data scientists to be able to identify drugs but also to analyze data, trial methodologists and statisticians for innovative approaches in clinical trial design, and neurologists and clinical trialists, as well as working together with funders and charities from across the United Kingdom. And it's really an exciting time for thinking about identifying new treatments for this condition. There are a number of better models of disease, either animal models or high throughput stem cell screening assays. Also, using AI and machine learning, you can use computational approaches for drug screening, and there's a growing body of literature, and AI can also be applied to the literature searches for identifying new treatments. We've adopted a new approach to clinical trials in neurological disorders. It's called a multi-arm, multi-stage adaptive platform trial. Now, it's not a new approach in trials in general. The first big MAMS platform trial was in 2005, the STAMPEDE study for prostate cancer. And then there have been other studies. The first study in neurological diseases was the MS-SMART trial in 2013 for progressive MS, multiple sclerosis. We launched in 2020, and shortly after us, the COVID pandemic started, and the recovery trial is one of the seminal trials in COVID-19 that led to improved outcomes in this condition. And in the last 2 to 3 years, there have been a whole host of multi-arm, multi-stage trials for other neurological disorders, including Parkinson's disease and Alzheimer's disease. And this approach has a number of efficiencies. You can test multiple treatments simultaneously against a contemporaneous standard of care or control. So that means that you can test multiple treatments faster, the cost is less, and the participant number is less as well. It's also a flexible platform in that you can bring new drugs in when the evidence base is there to test them. So if you think about a traditional two-arm trial design where you're testing one drug against a dummy drug or placebo, if you were to test one drug at a time, by the time you've identified the drug, started to test the drug in different sites across centres, collected the data, cleaned the data, and then analysed the data and reported on the result, it will take at least 4 years from first identifying the drug to having a definitive result, usually longer. And if you tested 4 drugs in sequence in this way, it would take the best part of 20 years to identify that those drugs are working or not. In MND-SMART, as I mentioned, we're testing many drugs simultaneously, and this has already demonstrated to be a much more efficient and quicker way of testing medicines than the traditional two-arm approach. One of the approaches that we're using to identify new medicines that I wanted to talk about are high-throughput, human-based stem cell screening models. We know a lot more about the underlying cause of disease now, and we have better models, particularly a stem cell model, which can look at replicating how abnormal neurons and supporting cells look like in a dish. And we can screen drugs at pace using robotic approaches, and whole libraries of drugs can be tested to look at reducing the abnormal protein that accumulates in MND, TDP43, also the effects of motor neuron excitotoxicity, how things are working on the supporting cells, the astrocytes and the microglia. Using this approach, one example of a drug we're currently testing in MND-SMART is a drug called tacrolimus. Tacrolimus is a drug that is used in transplant medicine. We're using this at a much lower dose in the clinical trial after evidence from our stem cell screening assays that this drug reduces the abnormal misfolded protein, TDP43. It reduces astrocyte reactivity and inflammation in the supporting cells, the microglia and the glia. It also reduces abnormal pathology in an animal model, a mouse model of the disease. So the design here is a summary of where we are with MND-SMART. We launched in 2020. We started with 2 repurposed medicines, memantine repurposed from dementia, trazodone repurposed from depression. Memantine dampens down excitotoxicity, and trazodone reduces stress granule formulation and abnormal accumulation of abnormal proteins. Both those drugs were found to be ineffective. They did not improve functioning in MND or improve survival. And it was important for the community that we reported on those results quickly. We introduced amantadine, a third drug, into the platform in April 2023. And because the platform was already established, we could launch that drug simultaneously across all of the sites that were open at that time. That drug passed stage 1 interim analysis and is now approaching stage 2 interim analysis. And this spring, we launched tacrolimus, the drug that I just mentioned now. And overall, we're looking at survival, the impact of these drugs and how long people live for, but we're also looking at functioning using a clinical rating scale, the ALS-FRS, and we're measuring changes in blood-based biomarkers. So we're looking at changes in neurofilament light chain and the impact of these drugs on memory, on mood, on quality of life, and on safety. Since our launch in 2020, we have randomized nearly now 1,000 participants across the whole of the United Kingdom. I mentioned that 2 drugs have been definitively tested, and we have new sites opening still across the country. I mentioned it's important to report on results definitively so that these drugs don't need to be tested in this population again. One of the criticisms of previous trials in ALS is that the same drugs have been tested again and again because the number of people studied has been small and the duration of follow-up has not been long enough. This study for memantine and trazodone, the first 2 drugs we tested in the trial, included 530 people, almost 175 people on memantine, 175 people on trazodone, and 180 on placebo. What's important is that the characteristics of the population was very similar across all of the groups and generalizable to the wider MND population. Because we co-designed this study with people living with the condition, there were very few withdrawals. The rate of attrition was very low. And that's because we've designed every step of the way of the study alongside people living with ALS MND. And this is important because people living with this condition are often sidelined, and we understood that patient advisory groups are not a tick box exercise. People living with the condition bring a unique perspective, and most researchers can't replicate this experience. People living with the condition are experts of the disease, and they live with the condition every single day. And so we've been working closely together with people from the beginning on the study design, the drug selection, the way that the drug is administered, the different routes of administration, the dosing schedules, the use of placebo, looking at all of the participant-facing information, the regulatory submissions, even the grant submissions and the authorship of papers as they come out. And just here, summarizing some of the examples by which we have innovated in the trial. We consent for people remotely, we use electronic cards for monitoring compliance, we use outcome measures that are remote, we courier the drug to people's homes, and we use video conferencing based follow-ups. We're very keen to introduce more digital technologies as well. And whilst we have not yet found a treatment that improves outcomes in terms of survival or functioning, we have changed the conversation for people living with this condition from one of breaking bad news and then just being able to offer symptom control and end-of-life care to one where we now create hope and expectation and a sense that people are contributing to a community to improve outcomes for themselves and other people. One of the other benefits of a platform trial like this is that we are collecting hundreds of samples from people at different time points across their disease, and these samples are linked to a large amount of clinical data that's been collected in a structured and systematic way through questionnaires and other data. So we're collaborating with a number of different organizations to develop new blood tests for diagnosis and monitoring of these conditions by looking at abnormal proteins that may be identified in these conditions. And just two examples of this are a platform called Simoa and a platform called NewLISA, which can look at large numbers of proteins at pace from small amounts of blood. And some preliminary data from our studies have demonstrated that there is an elevation in certain proteins that can distinguish between healthy individuals and people who have MND-ALS. And we're now looking at more samples from the trial that have been collected over time to look at this, to see if we can develop better markers of the disease. One other thing we're very interested in doing is looking at digital outcomes, and we understand that speech can be used as a biomarker for disease. So we know that people with motor neuron disease have speech that is affected, and we are using an app to collect speech from participants and people living with MND in Edinburgh, and trying to understand if we can differentiate between healthy individuals and people with disease from the way that they speak rather than the content of the speech. And so far, preliminary results have shown that this may be a viable option in motor neuron disease. So, what about next steps? We have already identified a shortlist for our next drug, the fifth intervention, to launch in the trial. And we're very excited about the combination, prospect of combination therapies. We know for a complex disease like MND-ALS, where there are different pathologies that lead to symptoms, that combination of drugs may be better than drugs working on their own. And this has been particularly effective already in cancer, infectious diseases, and in other areas of medicine. So we think this is the right approach to take. I mentioned about implementing precision-based blood markers and also digital markers like speech as well. I mentioned that there are other trials that are operating in this area for multiple sclerosis, Parkinson's and Alzheimer's, And we work as part of a community alongside these trials to learn more about how to operationally deliver these studies and how to learn about making the platforms even more efficient moving into the future. We're also very excited about working now with colleagues in Europe, and we're hoping over the next 12 to 18 months to be able to open MND-SMART in some sites in European centres. In summary, what we're trying to do is to encourage more people to participate in clinical trials, but to use the clinical trials as well to help with the drug discovery and the identification of biomarkers. And ultimately what we're trying to do is to improve outcomes of people living with all of these conditions. Many people have been involved in this study over the last 5 years. I mentioned from different disciplines, including people living with the condition, all the way through to neurologists, drug discovery scientists, and clinical trialists. So I just wanted to end by thanking all of them and our funders as well. And thank you very much for your time.
All right, thank you so much, Dr. Pal. That was an excellent presentation. Thank you for sharing it. All right, so I have a couple of questions for you. The first one is related to, let's say the structure of the platform. Where we've been talking with people from the Healy platform, for example, and the Healy platform is more. They have explained us that the Healy platform is more of a phase 2 environment, right? Is it the same case for the MND-SMART, or you also perform phase 3?
Yeah, thank you. So MND-SMART is a seamless phase 2 to 3 platform. We have interim analysis points. So our first interim analysis is when 50 participants in each arm of the study have been followed up for 6 months. Stage 2 is when 100 participants have been followed up for 12 months. Stage 3 is when 150 participants have been followed up for 18 months. And stage 3 really is equivalent to a phase 3 because at that point we're looking at survival and also rate of change of ALS-FRS. And that's because the regulators in Europe, in the United Kingdom, do look at survival as the primary outcome that will define if a drug can enter for routine treatment and be licensed.
So you already mentioned that you are adding two new compounds, and they are combination therapies, as I understood from your presentation. So what can you tell us about those two new combination therapies, and also when can we expect next results on tacrolimus and amantadine?
So amantadine is reaching its second interim analysis point later this year. So we'll be able to report on whether the drug stops or continues at that point. Tacrolimus launched just a few weeks back in the United Kingdom, so we are just in the early phases of that trial. In terms of the next drugs, we have initially a single agent, two drugs in isolation to start with, but then our ambition is to launch a combination therapy within the next 18 months. And a combination therapy really identified from our drug screening platform, maybe thinking about drugs, different agents targeting different pathways. So one, for example, that might be reducing the abnormal protein accumulation, and another that might be reducing excitotoxicity or reducing inflammation, and using two in combination. Now, what's difficult with that is to make sure that the drugs don't interact with each other and have large exclusions with side effects. So that's something we're working through with our patient advisory group at the moment. But we will certainly be announcing information in the next 6 months about the first two drugs that we'll launch, and then the combination therapy will follow.
Excellent, thank you. And if someone who lives in Latin America, for example, wants to participate in this trial, someone living with ALS from Latin America maybe is interested in being part of the MND-SMART platform trial, is that possible, and how can they do that?
So we're very keen to open more sites, and I mentioned about our idea of opening globally and opening in Europe. We'd certainly be very keen to discuss opening the study in South America with neurologists and colleagues there who would be investigators for the study. It's very important to study populations across the world for different backgrounds to understand if these treatments are working across different populations. At the moment, we are really just looking at international expansion of the study from the United Kingdom, and certainly if there are neurologists in Argentina or other countries in South America who are interested in supporting and working alongside us, we'd be very keen to speak to them.
Wonderful, thank you. So, lastly, we recently talked to Dr. Al-Chalabi, and he told us about all the process that went through to finally gain access to this fantastic grant from the UK government, like something like 50 million pounds, and which gave way to the possibility of starting with this UK MND Institute. So how would you say this impacted, I guess, positively on ALS research in the UK? And on the other hand, we also been talking to colleagues from the US, from the USA, like Dr. Jinsey Andrews and Dr. Bedlack, and well, they are going through a very different landscape in that country because of all the budget cuts in science. And the second part of the question would be how do you think that could impact negatively on the rest of the world?
So I think that funding is very important for progress in this area. I think we are at a tipping point with more information being identified about the biology of the disease. That then feeds into better drug discovery, and the better drug discovery needs to feed through to clinical trials. And I mentioned our feeling is the most efficient way of delivering clinical trials is through platform trials. That requires sustainable funding. It's not for a fixed duration, but if you're launching a platform, we've been running for 5 years and hope to run for many, many more years, it requires investment. And thus far we've been very lucky and fortunate in the United Kingdom that there are a number of charity funders, research council funders, and you mentioned government have invested awareness and funding in this area as well. And I think that support is necessary to keep the disease in the spotlight and the unmet need in the spotlight, and therefore leveraging against those awareness can bring in more money. So our view is that yes, it does require secure investment, and the investment is in people, in infrastructure, and in drug delivery, drug discovery, biomarker discovery. All of these things are really important. I mentioned before that international collaborations are very important to try and answer these questions. Our view would be outward looking towards collaborations globally. And I think that, in response to your last comment, that reduction in investment and kind of inward-looking approaches risk slowing down progress in this area.
Great, very clear. Thank you, Dr. Pal. And thank you so much for sharing some time with us for the second time and giving us this excellent presentation. It's very good to have again some updates about the MND-SMART platform. This is very important information for all our community, also in Argentina and the Spanish-speaking countries. So thank you so much for that, and thank you for keep fighting for this cause and keep pushing to get effective treatments for our patients.
Okay, very good to speak to you always. Thank you very much.
Thank you, Dr. Pal. See you next time.
Thanks.
Excellent. Thank you.